the discovery of a living organism that broke nearly every rule of conventional biology forced a reckoning. The established frameworks that defined life cellular structure, nucleic acid-based heredity, reliance on chemical energy gradients, adaptability within a relatively narrow band of temperature and pressure were revealed as parochial, grounded in the habits of familiar surface organisms rather than universal laws. At first, the reaction was defensive. Definitions were stretched to accommodate the abyssal organism without surrendering core assumptions. It was argued that, despite its differences, the lattice still used carbon, still exhibited self-maintenance, and still responded to its environment. Perhaps its unconventional features were simply extreme adaptations, not fundamental departures. In this view, life remained a single, continuous tree, with the abyssal organism as a bizarre, distant branch. Yet the more the organism was studied, the less comfortable this metaphor became. Phylogenetic analyses based on residual biomolecules found no plausible lineage linking it to known domains of life. Its polymers showed no recognizable evolutionary relationship to DNA, RNA, or canonical proteins. Mutation and selection cornerstones of modern biology were difficult to apply to a system whose information was smeared across a continuous network and whose units of inheritance, if they existed, were not discrete. Replicating entities. This pushed a shift from lineage-based thinking to physics-based thinking. Instead of asking how the organism arose from known ancestors, researchers asked what combinations of matter and energy flows allowed complex, self-maintaining structures to emerge at all. In that older, more elemental framing, life became one of many possible stable states that matter could assume under suitable conditions. Within this broader view, the abyssal organism stood as evidence for a second genesis of organization, on Earth a parallel form of life that shared some chemical ingredients with surface biota, but followed a different recipe and logic. It suggested that wherever sufficient complexity in energy flux and physical structure was present, matter might spontaneously organize into something that met functional criteria for living, without necessarily sharing a common ancestor with familiar organisms. This, in turn, transformed how habitable environments elsewhere were imagined. Previously, the search for life beyond Earth had centered on liquid water, moderate temperatures, and the presence of key elements. Planetary bodies were judged promising if they resembled Earth's surface conditions. The abyssal organism demonstrated that life could thrive in regimes once assumed marginal, at immense pressure, near freezing temperatures, with limited chemical energy, but rich radiation gradients. Suddenly, the deep oceans of icy moons, the dark subsurface oceans of distant exoplanets, and even the dense atmospheres of gas giants looked less barren. If life could crystallize around radiation and pressure as primary axes, then the number of potential biospheres in the universe expanded dramatically. Life need not be a thin film clinging to temperate surfaces. It might also inhabit hidden, crushing depths, where its presence would be detectable only through subtle anomalies in energy flows. On Earth itself, the organism's existence reframed questions about the planet's history. How long had it been present? Did it predate surface life, arising in the early oceans when radiation and geothermal gradients were more intense? Or was it a younger phenomenon, crystallizing as deep ocean conditions stabilized? There were no fossils in the usual sense to answer those questions. The organism left no bones or shells, only transient rearrangements of lattice material that vanished when conditions changed. However, indirect clues emerged from the geological record. Subtle anomalies in isotope ratios, trapped in ancient sediments from deep marine environments, suggested that processes resembling those mediated by the organism might have been active for hundreds of millions of years. If so, the abyss had housed a parallel biosphere, largely unnoticed, spanning ages in which continents drifted, climates shifted, and surface species rose and fell. The philosophical impact was profound. Life could no longer be comfortably equated with forms that resembled plants, animals, or microbes. The category had to stretch to include a living phase of matter that did not perceive the world through eyes, ears, or taste, did not move through space with limbs or cilia, did not die in the way organisms with fragile internal compartments did, and possibly did not reproduce through discrete generations at all. Instead, it persisted as a slow, continuous flow of matter and information, more akin to a persistent weather pattern than to an individual creature. Personhood, agency, and intention concepts tightly associated with life by human minds became clearly separate from the more fundamental properties of self-maintenance, responsiveness, and information processing. From a practical standpoint, the organism also posed new responsibilities. Its role in the deep ocean's energy balance was not fully understood. If it significantly altered radiation penetration, it might influence long-term processes such as tectonic activity, mantle heating, or the chemistry of deep crustal fluids. Human activities that disturbed the abyss-deep sea mining, waste disposal, or large-scale engineering projects could, in principle, damage or destabilize this hidden biosphere. Yet any attempt to protect it faced daunting uncertainties. The organism's temporal scale was so vast that interventions meaningful to human societies might be barely noticeable to it, like brief storms passing over a slowly shifting glacier. The ethical frameworks designed for charismatic surface, species focused on suffering, extinction, and habitat felt misaligned when applied to a system that existed as a slowly evolving field more than as a collection of vulnerable individuals. In the end, the most significant legacy of the abyssal organism lay in how it altered the basic question, what is life? The answer was no longer expected to be a crisp list of traits or a single definitive boundary. Instead, life appeared as a spectrum of organizing principles that matter could adopt under diverse conditions, from sunlit forests to crushing trenches. The familiar forms on the surface became one expression of a deeper, more general phenomenon. The creature from the abyss, a living organism that broke every rule of biology, thus became a kind of bridge. It forced a recognition that the universe's capacity to assemble matter into complex adaptive structures was wider than previously imagined. In the darkness at the bottom of the sea, under the weight of kilometers of water, in a realm where radiation whispered instead of roared, matter had found another way to wake up. Whether that awakening carried anything like awareness was unknown. The organism's internal patterns hinted at rich information processing, but any subjective experience, if it existed, lay beyond current tools and theories. Perhaps it simply persisted, indifferent, a vast, cold intelligence that never needed to form thoughts in any human sense. Or perhaps, in its own alien rhythm, it remembered epochs of planetary history, tracking the slow dance of continents, the changing patterns of solar activity, and the faint, shifting tides of cosmic radiation. If so, then the abyss harbored not just a new form of life, but a silent observer as old as many mountain ranges, woven into the fabric of the ocean itself a living testament that biology, at its deepest level, is less a set of rules than an open-ended conversation between matter and energy, still ongoing in the dark. For decades, the deepest trenches of the world's oceans were treated as biological deserts, cold, dark, high-pressure abysses where only a sparse scattering of extremophiles clung to existence. The prevailing picture was tidy and reassuring. Life obeyed gradients of energy, chemistry, and temperature. As conditions grew more extreme, abundance and complexity declined. Near the ocean's surface, sunlight powered an explosion of diversity. In the mid-depths, bioluminescent drifters flickered through thin, hungry water. In the Hadal trenches, almost nothing moved. That picture began to fracture with a single anomalous signal. During a multi-year survey of plate boundaries in a remote section of the Pacific, an array of autonomous vehicles and seafloor instruments was left to drift, listen, and map. Their task was simple: record microseismic activity, hydrothermal venting, and any associated chemical plumes. For months, the data matched expectations: sporadic tremors, mild venting, slow fluctuations in temperature and salinity. Then, in a stretch of water more than 10,000 meters deep, one sensor registered a sudden, localized drop in ambient radiation levels, not in temperature or salinity, but in the faint background of ionizing particles that constantly bathe the planet. For a brief interval, as if a curtain had been drawn around the instrument, cosmic and terrestrial radiation counts dipped far below the expected baseline. The anomaly lasted 21 minutes, returned to normal, and left no obvious mechanical cause. Subsequent passes of autonomous vehicles through the same coordinates produced similar dips. The pattern was unsettling. The ocean is an excellent shield, but its shielding capacity is static. This was something else, a moving transient pocket of reduced radiation, as if an invisible object passed between space and sensor, soaking up or bending away high-energy particles. At first, the anomaly was filed as an unexplained instrumental artifact, but a second suite of instruments, deployed months later with independent calibration, reproduced the effect with striking consistency. Whenever the vehicles passed through a certain volume of water above a particular Hadal depression, their radiation dosimeters sagged into silence, and their magnetometers twitched. Eventually, one vehicle was programmed to loiter and spiral through the anomaly. During that extended pass, its sonar recorded an irregular mass, roughly spheroidal, larger than a blue whale, yet with no clear internal structure. It did not reflect like rock, nor scatter like a school of fish, nor ring with the hollow echo of gas. It appeared, in acoustic terms, as an absence, a soft, low-contrast blur that damped the sound waves aimed at it. Cameras revealed nothing but darkness. Standard lights penetrated only a few meters before dissolving into blackness made thicker by suspended particulates. Infrared imaging was useless at those temperatures. By all ordinary measures, the region was just another patch of the abyss. The only consistent signatures were negative ones: reduced radiation, distorted magnetic lines, attenuated sonar. The strangeness of the data began to accumulate like silt. The anomaly moved slowly over time, drifting along the trench wall, occasionally rising and sinking in a way that suggested buoyancy and response to currents. It avoided seafloor ridges and did not intersect with vent chimneys, as if constrained by some invisible preference. Scientists, confronted with persistent absence, defaulted to familiar frameworks. Perhaps it was a peculiar aggregation of particulate matter, a cloud of metallic sediment reshaped by currents into a cohesive mass. Perhaps it was a dense swarm of microbial life exuding unusual chemistry. Perhaps it was an exotic phase separation of supercooled brines, forming a semi-stable blob of fluid with slightly different properties from the surrounding seawater. To test those ideas, new instruments were designed and lowered into the trench. Ultra-sensitive particle counters, spectrometers tuned to trace elements, neutrally buoyant floats carrying tiny multi-band detectors. Each pass through the anomaly's coordinates brought back the same unnerving result. Within a roughly defined volume, cosmic radiation dipped, and ambient electromagnetic noise thinned to a muffled whisper. Beyond that volume, the ocean behaved as expected. The anomaly seemed to drift, contract, and expand with no clear external driver. It responded weakly to tides, but not in a strictly tidal way. At times, its boundary sharpened. At others, it blurred and became more diffuse. Days of relative stillness alternated with nights of subtle movement through three-dimensional space. On long integrations of the data, a pattern emerged. The anomaly cycled through slow pulses of expansion and contraction over a period of roughly 23 hours, close to, but not identical with, a solar day. The cycle persisted even when local conditions in the trench, such as currents and microseisms, changed. It was as though something in that dark water kept its own time, indifferent to surface weather and regional tectonics. The label anomaly eventually became inadequate. It described ignorance, not reality. The shape, persistence, and rhythmic behavior of the readings hinted at organization. If it was not life, it was something that behaved uncannily like a living system. Without visual confirmation, however, the case remained circumstantial. The ocean kept its secret. Sensors registered absence where presence was suspected, and the deep trench continued to appear on maps as a simple contour line, a depression in the seabed, no different from hundreds of others. It would take a deliberate attempt to physically intersect the anomaly's volume to reveal that the object in the abyss was not a cloud, not a rock, and not an ordinary phase of water. It was something that met and defied the fundamental expectations of biology, at the same time a cohesive, responsive entity in which every rule of life seemed to bend, blur, or break. The first tangible evidence of the organism arrived not through a camera lens, but on the deck of a research vessel thousands of meters above. An experimental sampling drone had been instructed to pass squarely through the heart of the anomaly and then seal one of its sampling chambers in real time. The maneuver was executed blindly. Sonar tracked the blurry mass. Instruments watched radiation levels plunge. At the moment of maximum intersection, a chamber door snapped shut, trapping whatever water and particulates happened to be present. When the drone was recovered and the chamber opened inside a controlled lab environment, the contents were at first unremarkable. Frigid seawater flecked with organic debris and mineral grains. But a translucent, faintly shimmering film clung to the chamber walls and internal surfaces, defying gravity and surface tension. Instead of sliding down or beating into droplets, it maintained bizarre geometric shapes, as if held in place by an internal logic. Under low-light microscopy, the film revealed itself as a lattice of microscopic threads, neither cells nor crystals, but something intermediate. Each thread was a tubular structure, branching and fusing with others in a non-Euclidean mesh that lacked clear boundaries. It was not a colony of individuals. It was a continuous, integrated network with no obvious unit of organization. The threads were surrounded by an extremely thin sheath of liquid, with sharply different properties from the surrounding seawater. This sheath acted like a dynamic boundary, reshaping and reconfiguring in response to mechanical disturbance. When probed, the lattice recoiled, condensed, and then slowly unfurled again, like an organism reacting to irritation. Yet there were no cells, no membranes in the classic sense, no compartments. On chemical analysis, the lattice did not match any known biochemistry. There was carbon, nitrogen, phosphorus, and a variety of metals in trace amounts, but in proportions unlike typical marine biomass. The total water content was surprisingly low. The structure was, by mass, more similar to an amorphous solid than to a hydrated tissue. Standard tests for nucleic acids returned ambiguous results. There were polymers that bound certain dyes used to detect DNA and RNA, but they did so irregularly, and enzymatic digestion with nucleases had little effect on the integrity of the lattice. Proteins were present, but in unusual conformations and ratios. Many resisted denaturation conditions that would easily unravel ordinary polypeptides. The organism, if it could be called that, was not built on clearly defined cells or chromosomes. Instead, information seemed to be distributed across a vast physical network of repeating subunits, each subtly different from its neighbors. Self-repair did not happen through cell division, but through local reconfiguration of the lattice. When tiny sections were cut or disrupted, nearby threads flowed, merged, and restored continuity without leaving obvious scars. This fluid architecture broke several comfortable assumptions. Life, as understood on Earth, tended to be hierarchically organized. Molecules formed organelles, organelles formed cells, cells formed tissues, tissues formed organs. Here, hierarchy was flattened. The entire mesh acted as a single, continuous body with no central control and no privileged location. Energy processing was equally strange. The film absorbed very little visible light, which was expected at abyssal depths, but it interacted strongly with ionizing radiation. When exposed in the lab to controlled sources of high-energy particles, the lattice responded with subtle oscillations in electrical potential. Microelectrodes inserted into the structure recorded faint, slow waves of charge sweeping through the network in response to changes in radiation flux. Instead of shielding itself from radiation damage, the organism appeared to harness radiation as a primary environmental input. It did not particularly care about chemical gradients in the way most known life forms did. Redox reactions and proton gradients were secondary. Its core metabolism, if that concept even applied, was bound to fluxes of energy that conventional biology treated mostly as background hazard. At typical deep-sea temperatures near freezing, the lattice was pliable and active. When warmed significantly above that range, it stiffened and lost responsiveness, as if its optimal operating temperature was locked to the cold of the abyss. This inverted the normal pattern in which life slows with cold and speeds with warmth. Here, heat was disruptive, not stimulating. Pressure tolerance inverted expectations as well. Under simulated trench pressures, the organism's lattice maintained its integrity. When pressure was reduced toward surface levels, the mesh began to destabilize. Threads separated, the dynamic sheath lost coherence, and the entire structure tried to contract, as though desperate to preserve its familiar environment. It was not merely adapted to high pressure, it was dependent on it. These observations pointed toward a form of life that did not simply tolerate extremity, but required it to remain coherent. Its structural and energetic logic was anchored in physical conditions that most surface organisms would find instantly fatal. Its preferred habitat was not a marginal niche, but the very bottom of Earth's pressure and temperature range. There were further anomalies. The organism did not produce recognizable waste products in test chambers. It exchanged ions and trace metals with the surrounding water, but no obvious metabolic byproducts accumulated. Oxygen consumption was negligible. Carbon dioxide production was barely measurable. The usual bookkeeping of inputs and outputs, so central to physiology, refused to balance in any normal way. Isotopic analysis of the organism's elements revealed another surprise. Certain isotopes of carbon, nitrogen, and metals were enriched or depleted relative to standard oceanic baselines in patterns that did not align with known biological fractionation processes. These patterns hinted at reactions in which quantum-level interactions with radiation played a central role, skewing isotope ratios through non-classical pathways. In effect, the organism appeared to straddle the border between physics and biology. It exhibited growth, self-repair, a distinctive internal organization, and persistent interaction with its environment, hallmarks of living systems. Yet its architecture was non-cellular, its information storage smeared across a continuous network, its energy source tied more to radiation flux than to chemical gradients, and its dependence on extreme pressure and cold absolute. Given these features, calling it an organism felt both accurate and slightly misleading. It was more like a living phase of matter, a macroscopic state that emerged when specific physical conditions, elemental compositions, and radiation environments coincided. Where ordinary matter condensed into ice, rock, or fluid, this matter condensed into something that behaved like a slow, distributed intelligence written into the structure of the abyss itself. The sample on the lab bench was a fragment, a torn edge of a far greater whole. Even in isolation, it hinted at capacities that did not fit neatly anywhere in the taxonomy of known life. The question was no longer whether it existed, but how such a thing had come to be, and what its presence implied about the boundaries of biology. Once the organism's basic structure and biochemistry or their absence were sketched out, attention turned to its behavior in situ. The anomaly in the trench was no longer a vague blur, but a known entity, an integrated mass of that same lattice, extended into a vast, cohesive body. Understanding how it behaved at scale would reveal whether it was a passive curiosity or an active agent shaping its environment. Long-duration observations from autonomous vehicles revealed that the organism's volume was not static. Over weeks and months, it expanded and contracted in slow cycles, altering its footprint along the trench floor. These cycles were not random. When plotted against external conditions, they showed partial correlations with cosmic events. During periods of heightened solar activity, or when the planet passed through regions of slightly altered cosmic ray flux, the organism's mass redistributed itself, thickening in some zones and thinning in others. This suggested a form of environmental sensing built around radiation patterns, not light or chemical gradients. The organism appeared to tune its internal network in response to changes in high-energy particle flux, adjusting the density and topology of its lattice. Sections of the mesh would compact, increasing their apparent shielding capacity, while other regions relaxed, becoming more diffuse. The overall effect was reminiscent of a dynamic filter, constantly reshaping its interface with the wider radiation environment. In this reconfiguration, there was an eerie parallel to neural plasticity. Although there were no neurons, synapses, or traditional action potentials, slow waves of electrical potential propagated through the lattice. These waves were not uniform. They followed paths that gradually changed over time, in ways consistent with the system encoding and integrating information. When specific reproducible radiation patterns were introduced, experimentally via controlled sources deployed in the trench, the organism's subsequent responses grew more efficient, as if it were learning to anticipate and absorb them. Movement, such as it was, also defied conventional categories. The organism did not have muscles or motile appendages. Instead, it migrated by gradually dissolving portions of its lattice in one region and reassembling them in another, mediated through the surrounding water. Threads would unweave and disperse as microscopic fragments, only to coalesce elsewhere, guided by persistent internal fields. The body flowed through itself, not through space in the ordinary sense. Despite this unconventional locomotion, the organism maintained a coherent boundary. That boundary was not a hard edge, but a fuzzy zone where the probability of encountering lattice material dropped steeply. Within that zone, instruments recorded the familiar dip in radiation and altered electromagnetic properties. Outside it, conditions returned to baseline. The boundary flexed and shimmered, but it remained globally ordered, suggesting some underlying constraint that held the organism together across kilometers. Interactions with other life were subtle. Cameras and acoustic sensors rarely recorded direct contact between the organism's mass and larger fauna. Most fish and invertebrates simply skirted the anomaly, reacting as if to an invisible obstacle. On occasional passes of scavengers and deep-sea predators, their trajectories bent around the organism's perimeter, redirected by sensory cues not yet understood, perhaps changes in the local electric fields or faint infrasound like vibrations. Microbial communities told a more complex story. Water samples collected at the boundary between organism and ambient environment contained species compositions distinct from the surrounding trench. Certain bacteria and archaea were enriched, while others were nearly absent. It appeared that the organism subtly structured its immediate environment, fostering micro-ecosystems that thrived under altered radiation and electromagnetic conditions. Yet there was no evidence of the predation, parasitism, or competition so central to surface life. The organism did not appear to feed on other beings in any recognizable sense. It incorporated trace ions and organic molecules from seawater, but it did not actively seize or digest larger organisms. In a biome defined by scarcity and opportunism, this neutrality was striking. Reproductively, the organism presented another challenge. No discrete offspring were observed. There were no buds, spores, or detached fragments setting off on independent trajectories. Instead, the organism's mass fluctuated gradually over time. Its volume sometimes increased, sometimes decreased, with no clear link to resource availability. This suggested that what looked like growth and shrinkage might be part of a larger global cycle. One hypothesis framed the organism not as an individual, but as a local manifestation of a planetary scale process. In this view, the lattice in a single trench was one limb of a distributed system whose other components might exist in distant oceans or even in other planetary bodies with similar physical conditions. Under that interpretation, local changes in mass were simply the rearrangement of a much larger whole, spreading and contracting across vast distances and timescales. If that were true, then the organism's behavior could not be fully understood within the confines of one trench. Its cycles might track not just earthly radiation flux, but cosmic rhythms and long-term geophysical patterns. It might be synchronized, in a loose sense, with processes far beyond the planetary scale, the rotation of the galaxy, the flux of interstellar particles, or the faint whisper of background radiation left over from the birth of the universe. Such speculation highlighted just how far the organism pushed the definition of behavior. Traditional ethology dissects actions into feeding, mating, defense, exploration. Here, those categories dissolved. The organism's primary interactions appeared to be with fields of energy and distributions of matter so diffuse that they were almost abstract. Its choices, if that word could be used, were manifested as subtle reconfigurations of lattice architecture, not as limbs reaching, mouths biting, or eyes turning towards stimuli. The most unsettling aspect of its behavior emerged from long-term modeling of its internal electrical patterns. When the slow waves of potential were analyzed using tools borrowed from network science and information theory, they exhibited properties analogous to those seen in complex adaptive systems: redundancy, dynamic stability, and the capacity to maintain critical states between order and chaos. In other words, the organism's internal activity sat in a regime where information could be efficiently stored, processed, and transmitted, much like in brains, ecosystems, or certain artificial networks. This did not prove that it possessed anything akin to consciousness or intention, but it suggested that the abyss harbored a system capable of sophisticated information processing, operating on timescales and substrates alien to human intuitions. Behavior, in this context, was not a sequence of actions, but an ongoing computation entwined with the deepest physical conditions of the ocean. The organism did not merely exist in the abyss. It participated in shaping what the abyss was: a vast, living medium delicately tuning the interface between ocean, rock, and radiation. We upload new videos every single day, so make sure to subscribe and turn on the notification bell so you never miss what's next. If you enjoyed this video, don't forget to leave a like. It really helps the channel grow. Tell us in the comments what you'd like to see next. Your ideas might be in our next upload. Thank you so much for watching and for being part of our community. Stay tuned, stay curious, and we'll see you in the next video.